hospital, they don't just look at hematocrit, hemoglobin. I mean, generally they always look for that, but they look for more than just that. They look for a lot of times electrolytes, like your sodium balance, potassium balance. They look at your clotting time, you know, all those other things. So blood is very important. Blood tells you so much about the health of somebody. And if you're really good, you can figure a lot of things out that could be wrong with a person by knowing what's in their blood. I hope this works. I used to have a white piece of cardboard that I put up here, and that was my screen. And finally they put this in, which is nice. But I can't get used to it because it's in the middle, and I always had it off to the side. And I also used to have a really tiny projector, but the bulb burned out and they didn't have a bulb to replace it, so I have this huge honking machine now. But it's better than nothing.
I feel like we're about to watch a video from like the 60s on how to drive safely.
I know, right?
Like a driver's ed video.
No, you know what though, this does come in handy. When we get to blood typing next week, I'm hoping, we're gonna play a blood typing game. We'll be able to do it online, we're gonna play it together and do blood typing. That's my favorite lab. Okay, so all this stuff that we're gonna talk about, which we're gonna be talking about again for the next 3 labs, all come from chapter 13 in the textbook. So if you're interested in reading more about this stuff, or you wanna look at the diagrams, definitely look at look at chapter 13. So this is where we're headed. We today are going to look at the functions and components of the circulatory system, and we'll talk about the composition of the blood, specifically the erythrocytes and what's in the plasma. We're going to look at leukocytes next week, and we'll talk about the different types of leukocytes and how they appear, what they do. We're going to talk about EPO, erythropoietin, which is involved with erythropoiesis. And then in the next lab, we're going to talk about blood typing. So next week on Tuesday, we'll be looking at the white blood cells, talking about erythropoiesis and leukopoiesis. We'll talk about blood clotting, hemostasis, which basically these last 3 points are about that. And then on Thursday next week, we'll be doing the blood types. So again, like I said, we're gonna work through this PowerPoint a little bit at a time. Okay, so first part, the circulatory system. It is important for transporting respiratory gases, of course, carbon dioxide and oxygen. Something kind of interesting, you know, in the atmosphere that we live in and that we breathe, there's a lot more gas than what you think. There's not just oxygen and carbon dioxide, there's also nitrogen gas, and there are a couple of other types of gases. But our bodies don't have the capability to actually utilize those other gases because of how our red blood cells are structured in terms of the hemoglobin that's found within them. We can't use anything else. So a circulatory system, of course, includes the blood vessels, and the heart is part of transporting the blood through these blood vessels. But we do have transportation not just of CO2 and oxygen, but of nutrients and waste products, and also hormones and things that regulate other organs. The circulatory system actually acts with the kidneys to remove waste products from the blood. And it also helps to aid in temperature regulation, right? You get too hot, what happens? Your blood vessels dilate and you do sweat. Where does that sweat come from? It actually comes from the plasma of the blood. That's where it comes from. So you sweat out some of that. And of course, when the blood vessels dilate, they tend to allow heat to evaporate. Whenever you're cold, the blood vessels constrict to keep the heat inside Also, blood clotting, as we said before, platelets are very important for this process. We're gonna be talking a lot about that on Tuesday. And also an immune function, which we'll be talking about on Tuesday, because the white blood cells are there, which have a very important immune function. So as I said before, the circulatory system really doesn't act on its own. It's part of the cardiovascular system, right? The heart makes a complete circuit by pumping the blood from the heart to all the areas of the body it needs to go, and then back to the heart again. So it is a circuit, a complete circuit made up of the heart and blood vessels. And then the lesser known, bless you, the lesser known of the systems that a lot of times we don't really get to as much as we should. It's a very important system though, the lymphatic system. The lymphatic system is again, it's kind of special because even though it is kind of lumped here with the circulatory system, it itself is not a complete circuit. You remember that from anatomy? The lymphatics begin where? It's okay, it's like riding a bike, you'll remember. Where do the lymphatics start?
In your lymph nodes?
In the lymph nodes? Well, lymph nodes are part of the lymphatic system.
Are the blood cells?
No, that would be in the blood, but you're getting warmer.
Not spinal cord.
Way cold. No, like way cold. No, it's actually the capillaries of the lymph vessels start at the blood capillaries. Okay, and then you have multiple parts to the lymphatic. It becomes capillaries, and you have larger and larger vessels, and then you have two main places where the lymphatics dump their fluid into the venous system at the subclavian vein. Right, you've got two major trunks, I believe, ducts that dump the lymph into the venous system. And that lymph is going to be from tissue fluid, it's going to be from the digestive system. You remember fats are not absorbed directly into the bloodstream initially. Fats are absorbed first into the lymphatic capillaries. Remember that? Known as lacteals. So basically the heart, as we said, pumps blood through the cardiovascular system. Blood vessels carry blood from the heart to cells and back. Of course, we have our heart, our elastic arteries, which give rise to muscular arteries, which give rise to smaller vessels known as arterioles, which drain into capillaries, which drain into venules, and then of course the major veins. And we start at a really high pressure at the heart in the aorta. What's our systolic pressure usually?
120.
Yeah, exactly, 120, right? So systole, it refers to the ventricles contracting, right? And the systolic pressure is the pressure that is in the aorta. Basically, that's what we test with our blood pressure, in the aorta at the time that the ventricle contracts. So generally, you want it to be about 120 millimeters of mercury, right? Blood pressure, your contracting pressure is about 120. Your resting pressure, your diastolic pressure should be about 80, right? So like 120 over 80. So you start out at 120 millimeters of mercury in the aorta, and when you make it all the way through back to the heart, guess what the pressure is now? In the vena cava, any idea what the pressure is? Pretty much 0. That's why you need the muscles pumping, you need the breathing, all that, those pressure changes to get, to get the blood back up to the heart because of that. So anyway, So the heart does pump the blood through the cardiovascular system, carries blood from the heart to all the cells and back. The lymphatic system has a couple of different functions. It does have an immune function in the lymph nodes. We have some lymphocytes, which are a type of white blood cell, among other things. So the lymphatics have an immune function. They also have a digestive function, because again, in the small intestines, the fats are going to be emulsified by the bile that comes from the gallbladder or the liver, going to go into the lacteals, which are little lymphatic capillaries, and then ultimately that fatty lymph will be dumped into the venous circuit up in the subclavian veins. It's not one thing, it's another. Let me see here. Okay, so, so there's that, and then the last function is actually for helping to pick up excess tissue fluid. So how does it do this? Okay, if this is our, say this is our capillary bed, okay, here's our arteriole. Okay, this is our venule side, and we'll say this is our capillary. So what happens is, on the arteriole side of the capillary bed, we have a high pressure, because we still have the beating pressure of the heart behind the blood on that arteriole side, right? So we have a higher pressure in the capillaries. So when you go from the arterioles into the capillary bed, it's like going from a big raging river into a lake, right? So even though the capillaries have really small diameter vessels, the total cross-sectional area is huge. It's like a lake. So essentially, when you go from the arterioles in the capillary bed, the pressure does drop substantially. But on the capillary side, the arterial side of the capillary bed, we have a high pressure that pushes water out of the plasma. And this pressure, we have a high hydrostatic pressure. Hydrostatic is like the pushing pressure. That pushes the water out of the bloodstream. Okay, and now it's out here in the tissue fluid. Now, on the venule side, we have a strong osmotic pressure, high osmotic pressure. So you know, we talked just briefly about osmosis. This is like a pulling pressure, because we're going to learn that in the capillaries, in the bloodstream, we have a lot of proteins and things that are in there. And those proteins, they're going to be attracting the water to come back into the capillaries. That's what osmotic pressure is about. So a lot of that water comes back into the capillaries. However, there is some water that stays out here in the tissue fluid. And if we didn't have a way to move it, what would happen is you would get all this pressure in your tissues. You know what it feels like if you're on your feet all day and you don't move a lot. You know, like when I used to hostess at Max and Irma's, like I would stand on my feet forever, right? And then what happened, like, you know, you start, it's not maybe like real noticeable, but you can tell that your ankles got a little bit bigger because of the water, right? Your socks indent a little bit or something. Well, that's where the lymphatics come into play, but the lymphatics don't have any pressure. So they really require your muscles to kind of help move the fluid. So what happens is the lymphatics, we have little lymphatics here, and they have little valves, which will open. These these are like little mini valves. They allow the water, to go into the lymphatic capillary. And in that way, the water can return back to the blood circulation. That makes sense? Anybody have any questions? So that's basically that part. Yes?
The association between like cardiovascular issues, high blood pressure issues, and the swelling of feet.
The swelling that comes from that?
Yeah. Like like if I go to the cardiologist, he wants to check to see if my legs are swollen, for an example. Like is that this process that you're explaining? Why that might be?
Well, yes, in part, but depending upon what kind of cardiovascular problem you have, if you have high blood pressure that results in like left-sided congestive heart failure or something like that, or right-sided congestive heart failure. For example, like if say you have right-sided congestive heart failure, the right ventricle, most directly receives the blood from the upper and the lower body, right? So if the right ventricle is not sufficiently pumping the blood, then the blood backs up, right? It'll back up in the circuit, and so what happens is that there's a higher hydrostatic pressure in the vessels because of the backup. So because of that, you're going to have a lot of excess water that's in the tissues, and maybe the lymph can't keep up with it. So that's why, so one of the signs that somebody has right-sided congestive heart failure might be edema, like even ascites, which is like a large protruding abdomen because there's fluid buildup, or swelling in the legs, swelling in the arms, things like that. So yeah, so it's sort of related.
Okay, thank you.
What you're saying, yeah. Okay. Any other questions about that? Alrighty, so moving on. Composition of the blood. So blood has formed elements, of course, which are the cells, both erythrocytes, or red blood cells, and leukocytes. And then we also have another formed element, which isn't a cell, which are the platelets. So here we can see some white blood cells, and these are the red blood cells. And these little specks, like we saw on the slide on Tuesday, these are the platelets. And then, of course, they're suspended in this plasma. And plasma isn't just water. There's a lot of stuff in plasma as well that we're going to talk about in our last slide. So when it's centrifuged, and this is basically getting into talking about our hematocrit readings we're going to be doing today. When you centrifuge blood, so what they do, they take a sample of blood, they put it in a tube that has an anticoagulant coating so that the blood won't clot. Because when blood comes in contact with glass, the glass surface, it will trigger clotting. So they use something like heparin on the tube, and that keeps the blood from clotting. And we put this tube into centrifuge, and through centripetal force, we end up pulling the heaviest components of the blood down. We separate out the components of the blood. So the heaviest component would be the red blood cells. And the red blood cell percentage is generally about 45% on average between men and women. It's about 45% on average. There is a range for men and for women, but 45% is sort of the middle. Then the next heaviest component is very, very, very thin. It's known as the buffy coat. It's like a little thin white layer, and it's so small. But this white layer is going to be all of your white blood cells and platelets, which make up less than 1% of the total components of blood. The largest and lightest component is the straw-colored plasma, which will sit at the top, and it's about 55% of the blood. So when we are looking at the hematocrit reading, specifically what we're looking at is the percentage of red blood cells in a centrifuge blood sample. That is what the definition of hematocrit is. So on the handout that you have, if you took a look at the first page, under activity 1, it says hematocrit. It says normal hematocrit for females is 36 to 46%. Normal hematocrit for males is between about 41 to 53%. So that's what I said. It's an average. 45% is about average for people. But that's what we're looking for. And this can tell us, like, if a person is anemic or if they're polycythemic, which we're going to define. Anemia, of course, means that maybe you're not making enough red blood cells or you don't have enough. You don't have enough ability to carry oxygen. Polycythemia means that you have too many, and that can be a problem as well. Thank goodness I'm almost done with this part. And the thing is, I would put in that information, but it doesn't stop it. It still comes up. I'll put the information in and it still comes up. All right, anyway, the total blood volume is about 5 liters of blood. It's like a couple of gallons of basically water or something. But interestingly, your heart every minute pumps almost all of this blood through your whole body. It's like 4.7 liters, I think, that your heart pumps through every minute. It's really amazing when you think about what the heart does. Now the plasma, as I said, is straw colored. And the reason is because it doesn't just include water, it also has ions in it, metabolites like urea, again, or ketones like we were talking about, hormones, and antibodies. Just in case there's something foreign that comes into our bodies, we're ready. Red blood cells comprise, again, most of the formed elements. As I said, you looked at that blood smear. What did you see mostly? Little pink circles, right? Because all the red blood cells. They do make up most of those formed elements. And as I said, hematocrit can tell us a little piece of information if a person is anemic maybe, or if they're polycythemic. That and other tests as well have to corroborate. In the plasma, which again is a large component of blood, 55%. It has plasma proteins that are associated with it, which make up about 7 to 9% of the plasma. We have albumins, which are the most abundant, 60 to 80%. Albumins, if you remember over here, we were talking about the osmotic pressure. And I said we have all these proteins that help to draw the water back in. The albumins are those proteins which create colloidal osmotic pressure that draws water from the fluid back into the capillaries. Sometimes we just call these plasma proteins, PP for short, but it's really the albumins. There's actually a lot of PPs in this course. What?
Body humor. It was just body humor.
Body humor?
Potty humor? Like
Body?
trash humor? You said there's a lot of peepees in this class. We all giggled like children.
No, but now when I think about that, that wasn't exactly, I think perhaps what I just said could be taken in other ways probably that were not meant to be that. What I meant was PT, those two letters, we use a lot, like the posterior pituitary plasma. So you know I'm not gonna say something.
We understood because.
Yeah, but you guys took it. You took it there, right?
Yeah.
I didn't, but you did. Okay, got it. But yeah, actually now that I think about it, man, I gotta like think about what I said. It's like the one time I think I was, oh, never mind, forget it. Anyway, it's good sometimes not to relive past faux pas. Alright, so anyhow, another type of protein are the globulins. They carry lipids. So these are things like your high density lipoproteins or your low density lipoproteins. LDLs. Those are the globulins. We have another type of globulin that's a gamma globulin. These are the antibodies. And then another protein, which we're talking about on Tuesday, is fibrinogen, which is very important for clotting. So we will be talking about that. And finally, when we do talk about clotting, we're going to say that after a clot has formed, actually that fibrin is going to squeeze the clot, and all of the fluid that comes out is basically the serum that's left over.
Like I said, okay.
Okay. Any other questions? Any question, or I should say, any questions? Okay, that is it.
Is there any quiz on this on Tuesday?
No, we're gonna, Tuesday, we're just gonna do the connective tissue, so lab 2. That's gonna be that quiz. We're not gonna do this yet. Okay, so, oh, wait, I do have to finish up with this slide, I forgot. This is it. Okay, so last slide, we have red blood cells. I told you last time that they are biconcave. They have a dip in the top and a dip in the bottom to increase the surface area for diffusion of oxygen and carbon dioxide. Interestingly, red blood cells do not have a nucleus, so what can't they do? They cannot divide, exactly. Yes, you did. You need a nucleus because you need DNA, right? In order for a cell to divide. Red blood cells can't do that. So they can't make a copy of themselves like other cells can. They don't have mitochondria, so they can't make ATP through aerobic respiration, which is okay because pretty much red blood cells just kind of float around the lazy river of life, right? Like in your body. They don't do much. They're just pretty much big gas bags is all they are. They kind of float through. The heart takes them, the muscles take them where they need to go, right? Through the blood vessels. So, but it's interesting. Each red blood cell contains about 250 to 280 million hemoglobin molecules, which we're going to talk about. And there are about 300 billion red blood cells that are produced every day in your red bone marrow, which is pretty amazing when you think about it. So that is that part. Now we can take a look at the handout for today's lab, the printout for lab 3. And there. And we'll go over this part. So first part of this lab is all about the life and the death of the red blood cell. Its life, what it does, and how it goes, how it dies. And in lecture on Tuesday, we're going to look at the structure of hemoglobin even more closely because we're talking about proteins next. And hemoglobin is made up of a lot of proteins. So first of all, let's read through this together. The top first paragraph on page 1. They are biconcave in shape. Red blood cells contain a cytoskeleton. Its cell skeleton includes the protein spectrum. Spectrin is a very flexible protein, which is why red blood cells can make it through teeny tiny capillaries. They can do all kinds of gymnastics to flex themselves, go through, do all kinds of great stuff. When they're mature, red blood cells do not contain a nucleus, but instead they contain approximately 250 million hemoglobin molecules, each of which can hold 4 oxygen molecules. So 250 million times 4 is 1 billion molecules of oxygen. Now, considering the fact that normal red blood cell numbers, actual numbers, if you look at the last page, you'll see that in males, between 4.2 to 6 million per little millimeter cubed, okay, of blood is normal. 4.2 to 6 million for a male. For females, 3.6 to 5.6 million. When you consider this, and each one of those little red blood cells has a billion oxygen molecules, you think about it, that must mean we really need to use a lot of oxygen because that's a lot. Now, It's not to say that every single red blood cell is completely saturated, you know, every hemoglobin is completely saturated, which is why usually like we're at about 98% saturation, 99% or so. Now red blood cells, they are produced when a hormone called EPO or erythropoietin is released from the kidney. When oxygen levels are low, either because of low atmospheric oxygen or because of a decrease in red blood cell number, the kidneys release EPO into the bloodstream where it ultimately travels to the red bone marrow and stimulates stem cells found there to become erythrocytes. Okay, so here we have our, let's say, for example, let's say we're at high altitude, okay, and the air is thin. That means there's not as much oxygen up there, okay? So say we have low oxygen, okay, we're in like a, you know, like high altitude situation. Let's go. So we're hypoxic, we have low oxygen. Okay, we have something called hypoxia-inducing factor. Okay, it is normally degraded by the kidneys. However, when there's not enough oxygen available, HIF, or hypoxia-inducing factor, levels rise. Okay, so HIF, we have an increase in HIF, also known as hypoxia-inducing factor. Okay, we get an increase in HIF, which causes the kidneys to release EPO, erythropoietin. EPO goes to red bone marrow and our red blood cell numbers increase, which shuts off the initial stimulus, which is a negative feedback loop. So that is pretty much what happens. That's how we know to get new red blood cells. Now, red blood cells usually live about 120 days. When they become old, the hemoglobin begins to degenerate, and immune cells called macrophages are going to engulf or destroy the dying red blood cells in the spleen. So hemoglobin, if we were looking at it, hemoglobin basically has three major parts. It has the globin, it has the heme part, and then it has iron. So hemoglobin has 4 globin chains, which I'm going to just draw here simply as circles. Okay, and in the center of each globin chain, so this is globin part right here. In the center of each globin chain, we have a little ring-like structure, which is red in color. And it gives, ultimately, red blood cells the color that they have. This little structure is called heme. Heme is a red pigment. Then we have in the center, like a little jewel, we have a molecule of iron. And iron is very important for your blood because we can't make hemoglobin without it. What's so important about iron? Iron is going to bind to our O2. So if we don't have enough oxygen, we cannot, if we don't have enough iron, we cannot bind oxygen. So that's the importance of iron. You with me on that? what do you have a question about?
And education is very interesting.
What is?
Education.
I can take this so many different ways. I have no idea what you're talking about.
I'm just saying, everything is interesting that you're teaching.
Just now though, not earlier. Earlier it was boring because, but you know, this will help you guys because like when we talk about protein structure, you know, you'll at least have known a little bit about iron. But there is one really interesting thing about all of this. You know, right? You've seen, well, hopefully you haven't seen, but you've seen your blood. Like if somebody takes your blood, right? And that blood they're taking is venous blood, and you know it's kind of like a garnet color. If you ever saw arterial blood is actually a real scarlet color. And the reason why there's a difference is because arterial blood, of course, has more oxygen in it. And when oxygen is bound to the heme, the pigments, it actually changes how the heme reflects light, and it makes the blood a little different color. In venous blood, when you lose some of the oxygen, the heme changes its the way that it's conformed or its conformation, and it reflects light differently. So that's why you do actually see a difference in color between oxygenated and less oxygenated blood. So there you go. Okay, so what happens now? The red blood cell is dying. It's, you know, old, spectrum's worn down. It's getting stuck in the capillaries and the spleen. So each part, basically, what has to happen is the red blood cell has to be degraded. So the plasma membrane, it opens up and all the hemoglobin molecules come out. And of course, there are 250 million of them. What happens to the heme part? It says that again, I'm reading off the handout here, but it says that the the heme is salvaged for reuse. The rest of the heme is, the iron is salvaged for reuse. The body won't get rid of this iron if it doesn't have to. It holds on to the iron as long as it can. But the heme part, the pigment part, has to be broken down. The heme is degraded to yellow pigment bilirubin. The liver does this. The liver secretes bilirubin into the intestines, and the degraded pigment leaves the body in feces as stercobilin, which is a brown pigment. So heme goes from red to yellow to brown, and the brown is what gives your feces that color. So now you can laugh. That's a bit, but it's true. And then the rest of it, again, we have the globin chain, which is all protein. We're gonna break it apart into individual amino acids. What kind of reaction breaks things apart?
I got old timers.
Old timers?
Ends of lysis?
Yeah, it does. I'll give you a help. You use water to do this. Hydrolysis. Hydrolysis. Exactly. Good. So hydrolysis of the globin chain will give us our individual amino acids, and then those amino acids can be used to make other proteins. Okay. So, all right. So that is that story, and we are almost done talking.
Oh.
I know. You know what? Truthfully, this particular lab, I never like so much because there's just so much to explain and talk about. But I can't talk about it in a lecture because we don't have enough time because we have to get through all the other stuff. So that's why I do a little bit more in here. You're being patient with me. You're hanging in there. All right. So the hematocrit is the first thing we're gonna do. Well, I think. I don't know. I don't have my glasses on. I can't tell really what your faces look like. You might look like you want to murder me, but
This is a 1 credit.
Yes, it is. It's 1 credit. It's really not as much. When you go back and you study this, you'll see it's not as much as you think. It isn't. If you look in the textbook at how much we covered today, it's only a few pages. It's not as much as what it all seems. I promise you that. I just talk. All right, so anyway, so we're gonna do hematocrit today. We already talked a little bit about that. I'll show you how to do it when we're finished with all this. We're gonna do the hemoglobin readings also. Now, hemoglobin is a little bit different. Here we're looking at the color of the blood to tell us how much hemoglobin there is in a particular volume of blood. And why we look at the color is because the heme is, of course, the red pigment. If you have a light colored blood, right, that means you don't have as much hemoglobin in there. And that could mean, if it corroborates with your hematocrit reading, if the hematocrit reading is low, it could mean that you have anemia. You see? So again, they look at they try to corroborate those things. What are you doing over there?
I was just saying my head, yeah.
Yeah, but like real rigorously. I mean, I don't know, it didn't look right.
I'm a little off.
Oh, by the way, sorry, this isn't gonna sound good. You just made me think of something. If anybody in here, and usually people tell me this, but if anybody in here suffers from seizures, see, that's what I mean. But no, but I'm not, I wanna tell you, if anybody in here does, like seriously does, I would like to know, even if it's a petite mal seizure, you know, grand mal, whatever, I'd like to know, just, you know, let me know. I forgot to mention that in class before, because I've had people who've had seizures before, so, you know, and I didn't know for sure. Yeah, you just,
Yeah,
sorry.
I don't know.
I didn't know how else to work it in, because I'd probably forget to say something if I didn't say it right after that, but yeah, but anyhow, no, it's good for people to know. Okay, so yeah, hemoglobin, we're looking at the color, so it's a very simple test. Notice that for females, the normal range is 12 to 16 grams per deciliter. Actually, that's the correct, in terms of how we measure in units now, you can even just cross out the grams per 100 mil on there. Just know the range 12 to 16 grams per deciliter for females, males 13.5 to 7.5 grams per deciliter. It's important that you learn these values and that you also learn the units that go with the values. That's important to put all of it. Why do you think that both hematocrit and hemoglobin levels for males and females, why do you think that those levels are always higher in males?
Testosterone.
Yes, you remember that. Yes, it's testosterone, but as men age and they drop their testosterone levels, sometimes the hematocrit and hemoglobin levels can drop a little too, but they'll still be in range, they just won't be as high. Alrighty, so very last thing that we're gonna talk about, and then I'll show you how to do this, and if there's time, maybe there'll be time, we can try to do the red blood cell counts, but I have to run downstairs and get the sample. I don't want to put the cart before the horse. Last thing is pathologies associated with abnormal red blood cell counts. So there are two types: anemias, which is where you have too little erythrocytes, and there's polycythemia, where you have too much. So take a look at this. You'll see this is the second to last page in the lab handout. It says anemias are a set of conditions in which the oxygen carrying capacity of the blood is reduced. It's accompanied by fatigue, chills, shortness of breath, and paleness. And interestingly, it doesn't take much, when we're talking about these ranges, it doesn't take you much to go to outside that low end of the range for you to really get a lot of symptoms. Like, my father had colon cancer, and I remember we didn't know what was going on at the time exactly. I kind of thought it was cancer. We weren't sure exactly, and it was very tough to go to the doctor. But it got to the point where he would take a shower in the steam, and he would not be able to catch his breath. And my father, like, you know, goes on a treadmill, like he's an active person. He'd have to come out, sit down on his bed in the bedroom, and like he was just breathing so deep and heavy because he just didn't feel like he could ever get enough air. And it was because he was slightly anemic. I mean, he was just outside the low end of the range. It doesn't take much, really doesn't. So anyway, so the types, there are different ways that anemias can develop. One way is you can have an insufficient number of erythrocytes caused by hemorrhaging, right? So if you get an acute blood loss, maybe you get stabbed or something, God forbid, you know, or you hurt yourself, you cut yourself accidentally, or if you have chronic blood loss. So again, colon cancer, why did he have anemia? Because those polyps a lot of times rupture a little bit, you bleed, and then they heal, and then they rupture. So that's a chronic blood loss thing that kind of sneaks up on you. Hemolytic anemia is a situation where red blood cells rupture prematurely. So in this case, things like malaria, for example, people have malaria, which isn't that common, but if you have malaria, you do have part of the life cycle occurring within the red blood cell, and that causes the cell to actually rupture, so it will cause an anemia as well. Aplastic anemia, this is where you have destruction or inhibition of red bone marrow. What would cause that to happen? What would cause your bone marrow to be destroyed? Think of a medical intervention. This medical intervention can actually cause a person to be...
Chemotherapy.
Yes. Chemotherapy, right? Chemotherapy destroys part of your red bone marrow, so that's why people are more prone to infections, because the white blood cells can't be made. People get really tired, fatigued, because of the anemia that can result. So that would be an aplastic anemia.
Is that why they're at increased risk for more problems?
Why they're what?
Is that why they're at greater risk of getting cancer again or getting sick because of after chemotherapy because they lose red bone marrow?
Well, they not at being at higher risk of getting cancer, but definitely a high risk of becoming sick, yes, because, well, it takes time for the red bone marrow to build itself back up. But definitely during the time you're going through chemo, you're especially vulnerable. Yeah. Okay, so the low hemoglobin content is another way in which we can have anemia. What can cause this? Iron deficiency anemia. We don't have enough iron in the diet, so you can't make the hemoglobin because you need the iron for that. It carries around the oxygen. Also pernicious anemia. Pernicious anemia is where a person they can't absorb vitamin B12 because they lack something in the small intestine called intrinsic factor. And actually, this is sometimes found in older people, but younger people can get it too. And they usually give you an intramuscular injection of vitamin B12 because this way you don't have to have it pass through the digestive system. It's already in the system if you get a shot directly. And abnormal hemoglobin. So this is where the hemoglobin can't develop properly. In other words, you can't produce a normal hemoglobin. There are thalassemias, which is a class of conditions that make the globin chain absent or faulty, which obviously means that the hemoglobin molecule is not working properly. Signs of this are really thin and delicate red blood cells. And then sickle cell anemia. This is where we have a defective gene that codes for one amino acid substitution. It only takes one out of several hundred amino acids to cause this particular globin chain not to be forming properly, and that causes problems. Interesting thing about sickle cell anemia, it was actually sort of an evolutionary thing that helped people to not get malaria, who lived in Africa. And so people, what would happen is when the spirochete that caused malaria got into the red blood cell, it caused stress and it caused the cell to sickle, and so they couldn't complete their life cycle, and the person was essentially immune. And that's okay if you're a carrier, but if you aren't a carrier and you have no genes for normal hemoglobin production, that's a problem, right? And that's what makes it so hard. And there's really nothing that people can do except get blood transfusions, and you know that's pretty tough, because pretty much every couple months you would need that, because cells get stuck they're not flexible, they get stuck in the capillaries. People are more likely to get heart attacks and strokes and things, which isn't good. Okay, and lastly, polycythemias, that's where you have an excess of red blood cells, an increased thickness or the viscosity of the blood. There are a couple different things that will lead to polycythemia. There's polycythemia vera, where there is cancer of the red bone marrow, causing the cells to be produced, but they're not mature and they're not as functional as they need to be. So, this gives me the chills just talking about. So what they do usually for this is they have to siphon off the red blood cells. They have to siphon them off. They actually like drain some of them. And what they do when they take it out, if you ever look at it, it's like red yogurt. That's how thick it is. It's so thick when somebody has this type of cancer. It's just very, very thick. And of course that would be dangerous, because it could, again, make the blood too thick and heart attacks and strokes and so forth. Secondary polycythemia, this is when less oxygen is available at a high altitude, just like we talked about here, or when erythropoietin production increases, which it usually would at a high altitude or any type of hypoxic situation. And dehydration, because dehydration, this is a tricky one. If a person is dehydrated, it can actually make your hematocrit levels look like they're higher than what they are. So you have to be careful. Hematocrit and hemoglobin alone don't always tell you if a person has anemia or polycythemia. You also have to do a complete red blood cell count, which is what we're gonna do on Tuesday. And the reason for this is if a person is severely dehydrated, what component of the blood is gonna look like there's less of it?
Oh, nap time.
It's really raining.
I told you.
Is that what that is?
Rain.
It's pouring.
Good thing we're in class for another hour, huh?
Please.
Boy, you are like dripping with sarcasm, aren't you? You are like sarcasm, like central at you.
I thought I was being positive.
Kinda, sorta. No, all right. So, okay. So, what part of the plasma? Or, oh, I just answered it. I was gonna say the right. Oh, you did answer it.
Yeah, kinda. I mumbled it in case I was wrong. So
Oh, okay. Well,
It's a good technique.
It's a standard technique. It's a standard yeah. So anyway, so if a person's dehydrated, the plasma level can look like it's lower relative to the red blood cell part. So it could look like a person has polycythemia, but they really don't. So that one you have to be careful. And the last one is blood doping. And for this, I'm sure you've heard of blood doping. There's a couple different ways that you can do it. This, of course, causes a very unnatural polycythemia, because it's not because of a high altitude that you're doing this naturally. It's generally kind of an artificial situation that you set up. One way that's most commonly done now is people inject EPO, right? So you inject your own exogenously erythropoietin. That causes the red blood cells to increase in number. But of course that's illegal. I think that's what Lance Armstrong did, and that got him into trouble. The other way they do it's really gross. Again, they take the blood out. So they'll take blood out, they put it in a heparinized tube in a refrigerator a couple weeks before an event. Then your body does naturally make the red blood cells because you don't have enough. However, they'll take the blood that they stored and inject it back right before the event so that they have more blood. Now, of course, this is dangerous because, again, I mean, you dehydrate a little bit, your blood becomes thicker, and you do put yourself at risk of a really bad, you know, stroke or heart attack or something like that. It's not a good thing. So it's dangerous, but it's, again, it's also kind of gross. But I guess athletes depend on that. That's why a lot of times athletes do train at higher altitudes, because you just naturally will produce more red blood cells, so that if you're at a lower altitude, when you actually play in the event, you know, you're good.
You said like guys at the gym wear the masks when they're working out.
Yeah, I've seen that too, right? Same thing, good. All right, I'm done, I'm gonna shut up. So here's what we have. The last thing, I'm gonna kind of break into groups. This is gonna go very fast. Let's see. Okay, we're on 116.
Oh, time for you to.
No, I'm just trying to think. I really don't think we're gonna have enough time for red blood cell count, so we might be done early.
I'd rather go take a shot, like.
Take a shot of what?
I don't
What are we
know.
talking about here?
Hennessy.
I am gonna give this to you though, because this is a handout, we're gonna use it on Tuesday. So this is a handout for the red blood cell counts. But basically, I have a couple of samples you can take a look at that are hemoglobin
She gave me two
samples.
again.
We're gonna use a book to do this test, and then I have some samples for the hematocrits as well, and they're in these little pieces. And that's what I want to show you how to do. We can just break, I think if we break into two groups, as long as everybody can see, that's the key thing. How many do we have today? 1, 2, 3, 7, 8, 9, 10, 11. Okay, so like a group of 5 and a group of 6. So, okay, so let's do the hematocrit first. Let's break up, I'll do one group here and then do the other group. And if you're waiting for me to come around, if you want, you can try to figure out how to do this yourself. The instructions are written on there, but of course that will be covered up for the test.
Oh my God, this is crazy.
Not really. Okay, so I'm gonna go sit over here with you guys first. You guys want to turn around here and
Yeah.
Is it stopped? We're gonna keep it going.
Okay. So first of all, we get a sample. Doesn't matter which one. Am I being recorded again?
Yeah.
Well, I need to get like a manicure, and you know, if you're gonna do my hands, I gotta look real good. Okay.
Thanks.
Famous.
I don't know what I'd do without you guys.
So this is how the tube would be. This isn't real blood. I'm not allowed to use real blood in here because we don't do aseptic technique. I used to use real blood, and then they found out and they said no. So this would be like a tube of blood. They put it in a centrifuge and spin it out and put clay at the bottom, what this white is, so that the sample doesn't come out. And then what you're gonna do is take this. The first couple of steps are calibration steps, basically. You want the ratio of the red, right? So you don't want the clay in there in your ratio. So first you want to put the black line so it's between the red and the clay. You want to be right over it when you do it. And the second thing you do is we move this so that it's at 100. Okay, so we move that to 100. And then the swirled line is going to be kind of our upper level. We don't want the air in our ratio, right? So we're going to move the black line so it's in between the plasma and the air in the tube. So now we've got just the red and the yellow that we're going to get the ratio between. And then we take now both plates. So there's 3 steps we've done so far. We've taken out the clay, we set it to 100, and we took the air out, right? Now we take both trays and we rotate until we are right at this line between the red and the yellow. And then you can read right up here the number. And this number is about 50, I guess about 52%. That's all you do.
So what did we just calibrate? The plasma? Or what did we just figure out?
The
The
hematocrit.
red. Okay.
We figured out the ratio of the red to the yellow.
Okay. So the red is the 52%? Or is
Yes.
Okay.
Yeah, and if you look at it, it does look like the red is a little bit more than half when you look at how it is relative to the yellow. So, yeah, this